Okay, for this Essay 2 portion, or Exam 2, Essay 1, we're going to talk about the molecular characteristics of the contractile filaments found within the functional unit of the skeletal muscle cell. This functional unit is called the sarcomere. So there's a ton of these within the cell. So we have a ton of them. Just like this. Just kind of a quick drawing right here. But there's a ton of these different units. They're all called, this whole thing right here is called a sarcomere. This is what's doing the contracting. We're going to zoom in onto one of them, and I'll kind of talk about the different parts of it. So, right here, this is our sarcomere. This whole thing right here. It's a sarcomere. And then it's composed of these two Z discs. Z disc right here. And then we have nebulin up across here. Nebulin. And then we have titin, which is going to have our myosin heads or myosin chains on here. So we can go like this. And titin's kind of got this like tight, this kind of spring thing that helps with the contraction stuff. So it's called titin right here. And on this nebulin right here, we have F-actin, which is composed of a bunch of G-actin right here. And then it's got tropomyosin right there. So we can kind of blow up this little picture right here. So with F-actin, looks like this. It kind of goes around, kind of like an alpha helix almost along that nebulin. And it has these binding sites right here. And these are called, I'll do right here, our myosin binding site. Our binding site. And there's this thing called tropomyosin that goes along this F-actin and blocks those myosin binding sites. To talk a bit more about myosin, so we have a myosin head right here. And it goes out like this, actually a bit different. Like this, and another myosin head. And this is what's going on the titin there. There's these light chains right here, so myosin light chain. And then these are the heavy chains, which are including the head and then the tail as well. On this myosin head, there are different binding sites. So this is where it's going to bind to G-actin binding site. I'm going to just do BS. And then there's a different area right here for ATP to bind, which we'll talk a bit more later. So the goal of this myosin head is to be able to bind to the myosin binding site on the G-actin, but when there is the tropomyosin in the way, is unable to do so. That's where we need our buddy called troponin. And troponin is composed of a few different guys: troponin I, troponin T, and troponin C. Troponin I is in charge of binding to G-actin. And then troponin T is binding to tropomyosin. And then troponin C binds to calcium. Okay. So we have, our G-actin right here, right? It's got those myosin binding sites on it. But unfortunately, tropomyosin's blocking the way. And what we're trying to do in our contraction is to have the myosin head right here go and bind to that myosin binding site. But tropomyosin's in the way. So we have our troponin C that's binded there to it. So we have troponin I and troponin T that are binded to the G-actin and as well as the tropomyosin. Then we have just troponin C just hanging out right here. However, when calcium is brought into the intracellular, then that calcium, let's just do right here, calcium 2 plus, is able to go on and bind to troponin C. And what this does is it creates a conformational change, go like that, and actually moves troponin, sorry, I burped, tropomyosin out of the way so that our myosin head can bind there. Hopefully that makes sense with that. So this is what it looks like. So we have a bunch of tropomyosin right here on both sides. And these are what are going to reach up and bind to the G-actin. And then they'll do their power strokes and stuff, which we'll talk more about here in a second. But this is the functional unit of the sarcomere and the components of it. And I guess I could include the troponin that's always there. So when calcium comes in, it's able to have that contraction start, or that binding, so that contraction can occur. Okay, now we're going to talk about how we really get an action potential so that muscle contraction can occur. So we're going to start by looking at our neuromuscular junction, which is just the synapse here within a muscular or a muscle cell, right? So right here, I'm going to draw that. So we have our neuromuscular junction, which is pretty much this whole area, right? And I'm actually going to erase some of this here. Okay. So kind of like what we talked about before earlier in the unit, this is the kind of presynaptic terminal right here. And there's going to be a lot of vesicles that contain neurotransmitter. So here, and they have their snare proteins, right? These snare proteins need calcium to be able to come in and release that neurotransmitter into this synapse here. So what we have is that action potential is going to start kind of going down and opening up with our Wong right here, opening up sodium-gated, voltage-gated sodium channels right here, Na plus V. So sodium is going to rush in, and this is going to affect our voltage-gated calcium channels right here. And when those are have that Wong that comes by, they're going to open, and calcium is going to enter into the cell here, the neuron. This is a voltage-gated calcium channel. And this calcium will go down and open up, will attach to these snare proteins and allow a vesicle to release neurotransmitter into the system. And what we're going to have here are ligand-gated N1 receptors. So that neurotransmitter is going to bind to this, and we're going to get some sodium coming in, or whatever it is that we're needing. There's also like sodium channels here. So we're going to get that positive charge coming in, and it's going to start sending a Wong down. And we're going to have different sodium voltage-gated ions, sodium channels. So this is going to allow sodium to rush into the cell, continuing the whole thing. The calcium that we have that we're going to be using is going to actually be stored within the sarcoplasmic reticulum, which we can abbreviate with SARC right here. So we have a T-tubule right here. Yeah, you're good. And hold on. Okay, so we have our T-tubule right here. And in this T-tubule, there's going to be a lot of different kind of receptors down here. Like there could be potassium-gated channels. There can be calcium, chloride, kind of the same thing as up here. But it's a lot more concentrated inside this T-tubule, which is going to allow us to get to the sarcoplasmic reticulum. So it's going to keep going down, This is a sodium voltage gated channel. And we're going to reach something called DHP. And DHP is going to be closed until it reaches that Wang, right? So this is DHP. Okay. It's going to be connected to something called another receptor called RIRE. These are calcium channels. And RIRE is going to be opened by the DHP. So when that Wang comes, it's going to open. Both are going to open. And this one too. Okay. And inside the sarcoplasmic reticulum, there's a buildup of calcium inside here. And this, just to keep everything straight, this is the ICF right here. And then this is the ECF, including the T-tubule. That's still the ECF. So the calcium is going to be in here. Once this is opened, it's going to be able to leave the cell. And we actually have something really cool called calsequestrin that's binding to the RIRE. I always forget how to spell it, but calsequestrin. And this kind of brings the calcium ions like more around this opening, so they even rush out even faster. Because when we have an action potential for a muscle, we want to act quick, right? So this calsequestrin is going to allow that calcium to be pulled out a lot quicker. And the calcium is going to leave, and it doesn't actually leave like the ICF. It doesn't keep going out this way, right? It's going to actually stay within the ECF and go down this way. And within our ICF, we're going to have those basically the sarcomeres here, right? And this is going to be really important because on each of those sarcomeres, there's the the F-actin with the tropomyosin, troponin on that nebulin, right? And then our titin with a bunch of our myosin heads. And calcium is going to be really important for these, as we talked about earlier, for removing tropomyosin out of the way so that these myosin heads combine and have a contraction. So we're going to talk about that now here, kind of that little cycle. So we're going to focus on the myosin head. So we have calcium that's coming into our thing. I think I have room right here. I can do it. Okay. So we have our myosin head right here. Just like this. Right here, got our myosin head. And at this point, it has ATP on it, right? And it's been hydrolyzed, and ATP is going to bind. Let's just do it right here. Right here on that ATP binding site. And then we're going to have phosphate right here. And this straightens up the myosin head so it's 90-degree angle, right? We call this the Cox position. So it's pretty much how they're sitting, right? Until we get calcium that comes in. So calcium comes in and moves basically, if we want to draw out the whole thing, which I won't for the whole part here, has the tropomyosin here. It's in the way, but we have our troponin right here that's going to bind to that with that calcium, right? So I'm not going to include that. We're just going to assume that you understand that at this point. So calcium comes in, binds to troponin, moves tropomyosin, and then right here, now we are able to bind to that G-actin, right? So it's staying right here. And at this point, once that bond occurs, we release some energy. So we actually release some heat. And this heat causes for ATP and that phosphate. So ATP, sorry, I should have this here. So this is ATP. That ATP leaves, and same with that phosphate. And when those leave, when you have the phosphate there with the ADP, what happens is that you get a conformational change in your myosin, and this should be connected. So it ends up kind of dragging down, and it pulls along that nebulin, right, with the G-actin or F-actin, and there's some movement there. We call this the power stroke. That's where we're getting the contraction, and this is going to happen a ton. It's going to just keep going and going till like this starts going in and shrinking with our contraction with our muscle cell. That's why, like, when you flex your arm, it's like if it's stretched out, like your arm's just hanging out, and then you squeeze your bicep, you can actually see the bicep starting to shorten. That's exactly what's happening with the contraction. But obviously we don't want to stay contracted all the time, right? And these ones are going to move just a little bit. So we want to, when we're doing a contraction, like this cycle is going to happen a ton of times, but we don't always want to be stuck in contraction, right? So what we have is ATP comes in. ATP comes in and goes like this. It ends up binding to, the ATP binding site on the myosin head. And right now it's uncocked. So uncocked is when it's bent. I forgot how to spell it. But this is still uncocked. And, but then it'll go through hydrolyzation. So hydrolysis. And the ATP will turn in, I guess ATP should be a different color. So ATP is different from ADP. ATP, and this one right here is ADP. So hydrolyzes and separates into phosphate and ADP and is able to go in the cock position. And as long as calcium's located here, it'll just keep doing this. We'll just keep getting a contraction, more and more contraction, right? So really the way to stop this is to get rid of calcium from the intracellular membrane, right? So the way that we do that is we actually will actively pump it out through these things called circuit pumps. So these are circuit pumps. Okay. These require ATP, so energy, because we're going to be pumping calcium back into the sarcoplasmic reticulum, which has a higher concentration of calcium within it than the ICF. So it's going to be going against the gradient, and if you're ever going against the gradient, you need ATP, you need energy to do so because it's an active pump. So calcium will start leaving from here and go back into the sarcoplasmic reticulum so that we can stop our contraction because it won't be able to occur. Won't ever make that contact if there's no calcium. And these are the mechanisms behind pretty much an action potential in the sarcolemma, and how calcium is released, and then just kind of how like muscle contraction pretty much occur. Okay, now for this part 2 of our essay, we're going to talk more about the various sources of energy that we have for muscle contraction. So I can write them out right here. Let's see. So first we have cytosolic, ATP. Then we have creatine, phosphate. For all your gym bros, we have glycolysis. And then all these are anaerobic. We don't necessarily need oxygen to use these. And aerobic. And then 4, we have aerobic respiration. Okay. Cool. For this first one, cytosolic ATP. Basically, this is the pool of ATP that is readily available within the cytoplasm of the muscle cell. It's very small, kind of low amount of ATP here. But nice thing about it is that it's immediately available to use. Again, it's anaerobic, doesn't require any oxygen to make it. Really the only thing with it is that it's very short term, anywhere from like 1 to 5 seconds. It can really do activity. And this is something that you would see more so with like really short, quick bursts of activity, such as the muscle of the eye. So just like the eye. Then we have creatine phosphate. A lot of gym bros like to use this. And really it's just a high energy compound that can rapidly transfer into phosphate group from to ADP to replenish our ATP. And it's represented or facilitated by the enzyme creatine kinase. And kinase is something that adds a phosphate right there. And it's located in the M line of our sarcomere. And this is really messy now, so I'm going to erase some of this. Okay, cool. Its availability is a rapid energy source after like our cytosolic ATP sources have been run out. So once this is up, then we'll jump to creatine phosphate. And its advantage is really just a rapid way to regenerate ATP without using oxygen. But a disadvantage again is that it's very short term, for maybe about 10 seconds it can last. And it's good for short term, high intensity activities, just like weightlifting, like a normal set. It's maybe around like 10 to 20 seconds, maybe 30 seconds, right? It just depends what you're doing, but high intensity, right? Okay, then we have glycolysis. We all know a lot from biology, Bio 180. And it's basically the breakdown of glucose from glycogen mainly, that's stored in the muscle. And this provides a more significant amount of ATP than cytosolic ATP and creatine phosphate. So it's a pretty quick process, and it can function in the absence of oxygen and is a major source of ATP during intense activity. Disadvantage is it can only last for maybe about a minute before fatigue starts to, until you start running out of energy there. And then, so yeah, these ones are very short-lived without oxygen. And I guess, yeah, just be pretty muscular activity that's longer than a few seconds, but over a minute, that's when you start running out. It's kind of like when you're doing activities, like you can do stuff with holding your breath, but you reach a point where you can't do much. You need to breathe. So then this one, aerobic respiration, is occurring in the mitochondria. And if you remember, it's the powerhouse of the cell. It utilizes oxygen, O2, to produce ATP from glucose and from other fatty acids. And it can supply enough ATP to power the muscle cells for hours. So like I can just do hours right here. But it's a lot slower to start. So it's not like a quick access to get it, but it lasts for a long time. So it's not really great for intense activity, but it'd be for stuff like walking, jogging, riding a bike, just kind of stuff that's more, I guess, less demanding at that time. But yeah, I think that's it for this part of the essay. Okay, now we're going to talk a bit more about smooth muscle here. So there's two different types that we have. In the textbook, it says single unit smooth muscle and multi-unit smooth muscle. And really what it's referring to is phasic and tonic. We have these two different types, and most, I guess, muscle fibers, smooth muscle fibers, are composed of both phasic and tonic muscle cells, really, types of smooth muscle. The process for getting calcium is a little bit different than skeletal muscle, so we're just going to review that real quick here. So we have our cell right here, and it's got the smooth ER inside of it. Right here, smooth endoplasmic reticulum. Instead of the sarcoplasmic reticulum, it's just called the smooth ER. Perfect. And in these muscle cells, these smooth muscle cells, they don't have the T-tubules like they do in the skeletal muscles. So there's a bit of a different way to get this calcium that's inside the smooth ER to come out into the intracellular. So right now we're ECF, this is going to be ICF, and for a muscle contraction to occur, we need some sort of calcium to be in the ICF so that contraction can happen. So instead of the T-tubules with DHP, we're actually going to be using, let's see, this is going to be that RIRE. We're going to be using a G protein right here. So we'll have some sort of ligand. Normally it's going to be acetylcholine, and it's going to bind to one of our receptors that produce IP3. And this is the I forget the name of this one, but it produces IP3 right here. And some of these are going to be your alpha 1, M1, M3, and M5. We'll see if this one comes back to my brain. But anyways, this G protein has this little cascade, activates this. I think it's like phosphorylate C, something like that. And that releases IP3, and this IP3 is actually going to activate RIRE instead of DHP like in skeletal muscle cells. And this will cause it to open, allow calcium to rush out, and that's where we'll get this contraction to occur. So we're going to focus on right now on our phasic, because phasic is only dealing with the calcium within our smooth ER. And I'm going to move this out of the way, and hopefully I'm recording. Yeah, I think I'm recording. Okay, so we have that, and so kind of the same process as the skeletal muscle. So phasic's only using intracellular calcium. Okay, so we have our thing right here, our myosin head, and it's got, let's just do green for ADP with a phosphate on it. So right now it's in the cocked position, right? But in smooth muscle, there's no troponin. No troponin here that's able to bind to calcium, move the triple myosin, and allow the myosin head to bind to the actin, or the G-actin, right? So we have something else in place, and I'll draw it here. So we have something called MLCK, which kind of looks like a snake. It's kind of like this, like that. Cool. This is in its N-activated form, MLCK. Okay, and then we have comodulin right here. I'm hoping that's a name. I kind of forgot. It's like calsequestrin. I think it's called modulin. But it has 4 binding sites for calcium. So once calcium leaves the smooth ER and enters into the intracellular, then that calcium is going to bind to comodulin. So Ca+2, right? All that. And it's going to bind to MLCK and activate it. So then we get a form of MLCK like this. Okay, MLCK, and just go like that. Comodulin's activated right here. And what this is going to do is that MLCK can take ADP or ATP and hydrolyze it into ADP and a phosphate. And what it does with this phosphate is it actually throws it on to our myosin head, that large chain here. So let's just go like this. And that's what's actually going to be able to allow it to bind to the actin. So we have calcium coming in from the smooth ER. We have ATP that came in with MLCK. And it binds to the myosin so that it can now make contact with our actin here. Okay. Once it makes contact with the thing here, with actin, when the head makes contact with that, makes a bond, it's going to release some heat, kind of like what we saw with the skeletal muscle, right? It's going to release the ADP, so which was this before, and then it's going to release a phosphate. But it's going to keep this blue phosphate here that MLCK gave it. But it's still going to have a conformational change, but it'll stay connected. So there's our power stroke. It'll stay connected here, like that. But then something can happen here. So then we can get another ATP that comes in because it can bind easily to this when it's like that, and it'll go like this. Oops. It'll actually pull it off like that. You'll pull it off. This is ATP. Pull it off the actin right there. Here's our P. And then it can go one of two ways. So if that phosphate stays on here, it's going to stay and just keep doing this. It'll require no more ATP, at least for that case, right? Because it's binded there, so it's able to make contact with actin. But if we activate MLCP, which I can talk more about, which this is going to be hydrolysis, to put it in the cock position, is MLCP will come in and take away that phosphate group. So then it'll just go back to this. It won't be able to go back onto the actin because tropomyocin's in the way. Okay, so what MLCP is, it kind of looks like a Pac-Man, just like this. And I'm forgetting the name of it. Let's see. I'm going to check real quick. MLCP, rho kinase. Okay. So we have rho kinase that can deactivate this by putting on a phosphate onto him. So deactivate. And he's deactivated when there's basically a constant source of calcium. Because when calcium is a constant source, then rho kinase is activated and it can deactivate MLCP. And since in this case, we're only taking calcium from the smooth ER, there's a very limited amount in there. So we're going to, pretty much later on in the cycle, there's going to be enough calcium. It's going to activate, MLCP will be able to be activated, and then it'll take off the phosphate group. So we have a little chart right here. This is enzyme activity. And then right here is our calcium concentration. Okay, so let's just say right here we have calcium. So as levels get up, they'll go down because we run out in the smooth ER. But then we have, let's just say MLCK. As calcium is more coming in than MLCK can be activated by the calmodulin, and that'll start to increase. But we'll run out of calcium there. And then we'll have MLCP. It won't really become super activated because there's not like a constant source of calcium there. So it'll kind of start to come in, but these things will stop working anyway, so it won't stay very long. So this is for phasic. This is what you would see with like peristalsis, kind of like movement of food through the digestive tract. And yeah, so that'll take care of that. It's kind of in bursts almost, but it's still pretty slow. Doesn't take much energy. Tonic is a little bit different. So let's go back to this drawing right here. So it has access to the smooth ER, but it also has access to a greater source, which is in the ECF. There's a ton of calcium out here, and it's almost like an infinite amount because you'll never end up using it all. There's a ton of different channels that come in. There's some voltage gated ones. There's like, I don't know, sensory, like stretch receptor ones, mechanoreceptor ones. The idea is that there's a lot of receptors that can bring in calcium into here. And it'll give that more of the source. We also have a circuit pump here too that pumps it out of the ICF into the smooth ER, but it uses ATP. Same thing as skeletal muscle. And this is what's going to be giving us a ton more sustained calcium. And there's going to be so much calcium that circuit pump's not going to be able to do all of it. So there's actually a really cool channel that brings in some sodium and then pumps out to calcium right here. So that's kind of what we're dealing with. You just have a larger source of calcium. So it's ECF and ICF calcium. Okay, and it looks very similar to this here on the left. So we have our myosin head with our phosphate, and then this here. And then calcium's going to come in, activate MLCK. We're going to have some ATP MLCK is going to use and turn it into a phosphate, and it's going to plop it on our myosin here. And it's going to bind to the actin here. So phosphate, we got our ADP, and then we got our phosphate group. The thing is, now we've got sustained calcium, so what's going to happen is that urokinase is going to have that calcium, and it's going to be deactivated. So then this MLCP will be activated now, and it'll come in a lot earlier than it did here. So we're going to still release some heat. So heat, and then MLCP is going to come in, take off that blue phosphate here, the MLCK added, and we're going to lose with our ADP here too, and our other phosphate. So we lose a lot. We lose a lot here, and we're still going to have our power stroke right there. Cool. So that's going to happen, but it's going to be stuck on there. It's going to be stuck. And this is what we call our latch phase. So until ATP comes in to take it off, we're going to have this ATP trying to bind to it. But it's going to try, but it won't be able to until finally it is able to bind. And that's what's really nice about this is, like with sphincters, they can just stay closed for a long time without any energy because the ATP is not really able to bind because the phosphate's not there. And now it's really hard for it to fit with the actin there. So once it finally gets on there, we have a ATP on here, which is going to be this. And that's the end of the latch phase. And then it'll go into hydrolysis again. Right here. So our chart looks a little bit different now. Let's see. So now we have sustained calcium levels due to you accessing it from the ECF. We have our enzyme activity. And then we have, perfect, so our calcium. It's going to go up like this and then level off, it's sustained. And then we have our MLCK. There's enough calcium, MLCK will be happy the whole time. And then gradually with MLCP, as both are being there, it's going to start to catch up, be more active. But yeah, these are the main differences with the two different types of smooth muscle, basically, and phasic and tonic. Okay, now we'll just quickly compare and contrast smooth muscle and skeletal muscle. So smooth and skeletal. So big thing is that skeletal muscles are large and cylindrical, if that's the word, and I'll just do cylindrical. And they do contain like a multinucleated cell. Smooth are much smaller and have a single nucleus. Skeletal muscles are striated, and these ones are not striated. These ones have skeletal muscles have highly ordered sarcomeres, and smooth muscles have myofilaments that are quite a bit longer and not uniformly aligned, but they both have sarcomeres. This one has a T-tubule, and then this one does not. T-tubule, I just won't add it. Connective tissue, skeletal muscles have connective tissue, and smooth muscle really do not. Not as much, at least. Skeletal muscle is quick and powerful, and smooth muscle are long and sustained without fatiguing, which makes sense because we don't want like our bowels to like get tired, or even like, you know, what's holding our like our bladder and stuff, because they just got tired and like you're going to be peeing all over the place, right? Smooth muscle can contract up to 80% of its total resting length, and these ones can only do 30, so you really can't use a whole lot of your skeletal muscle. Let's see. Yeah, these ones do phasic and tonic contractions, and then this one does concentric, which is kind of stretching or contracting, concentric and eccentric. Okay, this one has troponin, and this one has MLCK that's able to put on the phosphate so that myosin head can bind. There's a latch state here on the smooth muscle, and that's why they can sustain a contraction for a lot longer. But really, yeah, they're both for different things. So like smooth muscle is more for your organs because you don't want your organs to tire out, but skeletal muscle is more for the muscles that you generally think of for powerful, rapid contractions. Now we're going to talk about more of the reflex stuff. So in our brain, we have, you know, our neuron that goes down. It's the upper motor neuron. And this is voluntary, right? And then it goes down the spinal cord right here. And then it connects to this one right here. This is the alpha motor neuron. Okay, and this is going to innervate a bunch of different muscles, right? So we have our muscles, and these ones are going to be called like muscle fibers. It's like this. They're all going to be innervated by this alpha motor. And we're going to call them, pretty much they're technically called extrafusal fibers. Okay. And then we have these things on them called the muscle spindle apparatus. And they're kind of like this on each one of these muscle fibers or extrafusal fibers. And they contain intrafusal fibers inside of them. Intrafusal fibers. These are going to deal more with stretch and stuff. So we have these that send back different signals. Neural like this. All the way back to the spinal cord. And they're called our sensory fibers. And we have different types. We have IA, 2, and 1B here. And then we also have right here, it's going across here to each of these, and they're called gamma motor neurons. Okay, let's blow up one of these muscle spindle apparatuses. We'll do right here. So it's kind of like this. Something like that. And we have two different types of intrafusal fibers. One that's kind of straight and one that's got a bit more of a bulge. This is our intrafusal fiber, and this one is our bag fiber. And each one of these inside is going to be, so this is for our chain. It's going to have that innervation right here for a static gamma neuron, and that's going to be coming in this way, right, going into it. And then it's going to be using number 2 sensory fiber. These have all their sarcomeres as well. These are spindles that basically help to keep tension to receive signals. So you need tension for these signals to be sent. And that kind of looks similar, but not for your bag fiber. So it's got these spindle fibers all up here in these thinner areas. And that's where you'll have, it coming in with the dynamic gamma motor neuron. Let's go to like this. Okay. And throughout this belly, it's going to have this IA right here, this sensory neuron. And they're both used for different things. So for this chain one, it's really for more kind of static stretching. So let's say that we increase our stretch right here, and it's going to start sending signals. It's like, okay, we're in this stretch position. It's kind of more for like duration. So this is more duration. But then for this one, if the same thing happened, it just detects changes pretty much. So if we have the same thing, it would only detect a change. So I'll send a lot of signals here, none in the middle, and then changes there. And this is more for pretty much like duration. And they both send signals based on stretch of this cell, or of these spindles and stuff, of the muscle. Let's see what else we want to talk about. Then we have our motor thing here. Our dorsal. This is our dorsal. This is our ventral. This receives sensory, and this kind of sends out actions. So this is pretty much what it looks like. Okay, so we get these signals that come in to like this right here, and they can go, so like say like we flex our quadricep, right? Then we'll get like a signal here to go right here and keep telling it to flex, right? So release neurotransmitter, like an ESPS or EPSP. And then it'll send that signal out to the antagonist or agonist, just pretty much saying to do the same motion, right? And these will be alpha. And then we also have an interneuron that'll be right here and pretty much inhibit the antagonist. They'll go out, antagonist. So we'll receive that signal, it'll tell the agonist to go, and then it'll inhibit the antagonist so that it doesn't flex. So that's pretty much with like your reflexes. Because if you had both your agonist and your antagonist going, we wouldn't move. Nothing would happen. So you need one going or the other. And if one's stretching too far, then it'll tell the other one to stop. We have the Golgi tendon organs too. So Golgi tendon. And it does the same thing pretty much. So we have like someone's leg, their foot coming up, and they got their leg. So let's just say they had their hamstring, and then you had your quadricep. So you have this tendon here, right here, and it's innervated with a Golgi tendon, which will go over to, let me draw it real quick. Like this. So we have each one innervated. So if I sense that this one is being stretched too much here on the quadricep, then this is your Golgi. Then it'll go here to an interneuron into the agonist, and it'll tell the agonist to antagonist to start pulling because there's too much stretch, so it'll start pushing the leg down. And then this one, your agonist, will be stopped to stop that stretching because it's like, okay, we're going too far, we're stretching too much, so we want to go back the other way. So I think I hit everything here. This one's kind of a weird question, but pretty much your Golgi tendons, I mean, kind of comparing your muscle spindles and Golgi tendon organs. Your muscle spindles are within skeletal muscle fibers, and then Golgi tendon organs are in tendons near muscle tendon junctions. And Golgi tendon organs have an inhibitory response, and muscle spindles have an excitatory. And muscle fibers are protective over stretch, which maintain muscle tone. And Golgi tendon organs prevent excessive tension on tendons. And then the stretch reflex and stuff, and withdrawal reflex, stimulus. I'm going to draw this so you know I'm here. Stimulus is muscle stretch reflex, and the withdrawal reflex is just pain. So it has a bunch of nociceptors, and it'll do flexion to the limb to withdraw from pain. So if like I'm getting pain, like right here, then you'll get a reflex to get away from that pain. And it's really meant to avoid pain and tissue damage. So your flexors will contract and your extensors will relax. Do that same thing. And yeah, so that's pretty much it. But yeah, that's it for this essay.